All right, welcome to the third episode of the Harry Matters podcast. Welcome back, Ralf. I think we showed you a couple of the memes that the Haired Out community was sending in. Couple people I think missed you more than they missed their girlfriends. Their words, not mine.
Oh, now I'm worried.
Um, they need their hair. Uh, so as just like a little overview, we had the reviewed by Ralph concepts, but I think you want to take a little bit more time, like really digging into the literature, maybe getting your written responses down, having a little bit of a back and forth with the community, and then hopping on a podcast to kind of go a little bit deeper into those.
Yeah, give me about a week to do that.
Okay. Sweet, sweet. So yeah, so I think this will be a bit more of like a casual conversation. I think there's some more generalizable things that people wanted to talk about. The first topic that we'll go into, and it's actually a little bit related to PAI-1 inhibition, but is mast cells and sort of their impact on the hair loss process. So I won't talk too much more. I'll let you sort of go into your experience with mast cells and sort of how you figure them into the whole process.
Yeah, so actually, my interest in these mast cells goes back to the time when I was a postdoc at Yale in the dermatology department, and so I had just started learning about hair follicle, and all our research was in the mouse, and I stumbled over a very old study from the 1960s out of Italy, where guy Giacometti, I think he was called, in Genoa, had found that when there is a hair cycle wave running through the skin of rabbits, there were massive changes in the numbers of mast cells and also in how much they degranulate, which is a sign of their activation. And it's mast cells, for those who don't know them, those are the cells that give you hay fever, urticaria, allergic asthma. So they are key inflammatory cells. They're also the only cell in your body that can kill you within a few minutes. If millions of them degranulate at the same time, you are dead meat if there is no emergency care available. So that fascinated me, particularly because my boss, my teacher in hair research at the time, Kurt Sten, was completely uninterested in immunology. He didn't think that was important in hair. And I thought, well, I like this. This is good. Let's look at it. And when I came back from Yale, I then did, in the mouse hair cycle, a very careful study where we, for the very first time, showed that each time the follicle switches from growth to regression, and then from resting telogen back into anagen, mast cells degranulate, and their numbers change during the hair cycle massively. We also could show that if you antagonize their products with antihistamines or serotonin antagonists, you can impact on the speed through which the follicle goes through the cycle. And if you make them degranulate, you give them so-called mast cell secretagogues that you inject into the skin. Among them, actually, some hormones, such as ACTH. We talked already about this in the context of hair follicle neuroendocrinology. So, turns out these mast cells, at least if you're a mouse, are massively changing in number and activity. And we published this 94 in Developmental Biology, and nobody gave a damn about it. I thought it was so exciting because it showed that these mast cells are actually, that was our hypothesis at the time, and I still think it's a fair hypothesis, that they are central switchboards of tissue remodeling, because mast cells have probably stored more different mediators, enzymes, cytokines, growth factors than any other cell, and they can make basically everything. And in addition, they are among the oldest immune cells, and they connect what happens in the epithelium and in the mesenchyme around them with more specialized immune cells like T cells. So that was nice, and at about the same time, and that really got me excited. About the same time, the famous Albert Kligman in Philadelphia at UPenn who was at the time one of the most important hair researchers and also a very famous dermatologist. In one of his last papers, he had published in '92, I think, in the British Journal of Dermatology, that when hair follicles, human hair follicles, that are about to get miniaturized, are moving from a terminal hair follicle via an intermediate hair follicle into a vellus, that in this intermediate stage, you find a lot of mast cells around them. Not so much that people had actually looked at it carefully, unless he did it. And that again was never followed up. And that chimed in well with what I had seen in the mouse. And then when I worked in Lübeck, at the University of Lübeck in Germany, I had a PhD student, Marta Bertolini at the time, who worked on mast cells in alopecia areata, in the most important inflammatory hair loss disorders, where again mast cells had been ignored more or less. Yeah, in some patients, particularly if they had if they were atopics, then they sometimes had more mast cells. But we actually noticed in this study, and published that 2014, that mast cells normally around the hair follicle are immunoinhibitory in human scalp skin. So they're actually good guys, under physiological circumstances, and they produce, they secrete substances, interleukin-10, TGF-beta, that keeps the immune system and inflammation under control around the follicle. But in alopecia areata, then we noticed, they completely become nasty. They become highly pro-inflammatory. They spew out all these mediators and aggressive enzymes, proteases such as tryptase. And they completely change their face and their function. And moreover, they start kissing CD8 T cells. So that made us believe, but we had no clear functional evidence, but this phenomenology made us believe mast cells can become quite nasty and important in at least selected inflammatory hair loss disorders. And we never target them. Yes, in these inflammatory hair loss disorders, we give cortisone, and that, of course, would also impact somehow on mast cells and inhibit them. But what the role of mast cells in androgenetic alopecia really is, ever since that study by Kligman, Jaworski and al., British Journal of Dermatology, 1992, I think, has not been followed up rigorously. And that is a shame, because we know how to target mast cells. We have drugs on how to target mast cells. There are products. We can directly up and down regulate even their number. This has been mainly made possible through great advances in the treatment of urticaria. This is a very nasty, excessive mast cell activity disease, where people get hives all over their body, sometimes over years. It was extremely difficult to treat, but now we have potent drugs that can either eliminate or downregulate mast cells. And if it turned out that mast cells are doing something that they should not be doing during androgenetic alopecia, particularly during the remodeling, the miniaturization of the terminal hair follicle into a tiny vellus follicle one, then we probably already have drugs that we could throw on there, topically, ideally, no? So again, that is in an area of hair loss research that is not aggressively enough pursued. We started the field. We didn't really pursue it ourselves, but if there's any interest in the community to do this and apply this to to mast cells in androgenetic alopecia, I'd be absolutely delighted. It's an old love of mine.
That's amazing. I think there could be plenty of there's definitely plenty of interest.
Oh, I forgot one. How could I forget this? So you remember that we talked briefly, I think, about the impact of psycho-emotional stress on hair loss.
Mm-hmm.
And that's where mast cells come in big time. Because mast cells are key cells in mediating so-called neurogenic skin inflammation. So when you get stressed, you release stress hormones and neuropeptides, stress-associated neuropeptides, to which mast cells are uniquely sensitive, or at least highly sensitive, and they start degranulating. And we had a mouse model that we published many years ago, where we stressed mice with sound. I think that's what we talked about. We put a beeping device into their cage, and that drove them crazy, and they got really stressed. And where mast cells come in there is, if you try the same thing with mice that do not have functional mast cells in their skin, there's no longer any hair growth inhibition and very little inflammation around the hair follicles that you otherwise see. So the neurogenic perifollicular skin inflammation that you can induce by severe psycho-emotional stress in mice requires the activity of mast cells. So any one of you who thinks, oh, my hair loss gets worse each time I'm seriously stressed by my girlfriend, by my wife, by my boss, by a test, by an exam, whatever, well, mast cells may then do something that they normally don't do. They then secrete lots of inflammatory mediators that across the board tend to be hair growth inhibitory. And that was all mice, but we also then found that if the key stress-associated neuropeptide, substance P, that is also operating in your scalp hair follicles, if we dump that on follicles, that's very strongly hair growth inhibitory, the mast cells degranulate, and so what we had found there in mice is probably quite well transferable to the human system. So mast cells also, not only are they central switchboards of tissue remodeling, and they are connector cells between so-called innate immunity and adaptive immunity in the immune system. They are coordinating immune responses, in other words. But they also connect you with your brain. And that's a scary thought, right? So one cell population that is specializing in connecting whatever impulses come from the sensory nerves in your skin, neurotransmitters being released, neuropeptides being released, mast cells see those, very sensitively respond to them, and translate them into secretory activities to which the hair follicle then also responds.
Wow.
Nobody works on this very seriously. And I think it's about time to do this in an androgenetic alopecia context.
Fantastic. So while you were studying mast cells, because a lot of our community members have been sending in articles, did you ever look at PAI1 and its relationship to sort of mast cell degranulation?
No, I have not done, and I'm guilty of not having done that. So it's actually an interesting connection. And I think there's even a mouse model out there where this is up or down, I think knocked out or overexpressed, I've forgotten. They have a hair phenotype. So it does make sense to look at mast cells again from context, but I have not done this myself.
No. Yeah, the theory I think that's leading right now is that endothelin-1, which obviously triggers dermal sheath contraction, is also a mast cell activator, and then also upregulates PI1 by about 90%, which I think.
But you know, mast cells respond to basically everything that's out there, and how much they respond depends on context, what other signals they see at the same time. So one has to be very careful with these cell culture studies where somebody has dumped substance A, let's say endothelin-1, on there and then seen the mast cells respond. That doesn't mean that they will respond in the same way in living human skin, because when they at the same time get 20 other signals, that will determine how they respond. Another thing that made me totally fascinated, and actually we discovered that. So there are many skin diseases where you have increased numbers of mast cells, in atopic dermatitis, in psoriasis, many in any kind of eczema. The number of mast cells goes up, and the immunologists, including my wife, who's a mast cell expert, keep telling us, well, these mast cells come from progenitors that are being flushed into the skin from the bone marrow. So they're hematopoietic progenitors. And yes, some of this may be true, but the really fascinating thing, and one compelling reason why one cannot ignore perifollicular mast cells, those that are located around the hair follicle, is that we proved that you don't need to have a bone marrow or any blood supply to get new mast cells in your skin, and they arise from progenitor cells that are already sitting there around the hair follicle in the connective tissue sheath of the hair follicle. So the follicle is basically having its own little bone marrow supply close by, from which it can generate mast cells whenever it needs them, and it can also suppress the generation of new mast cells. So when it makes what we proved is that hair follicles, when you stimulate them with corticotropin releasing hormone, the key stress response hormone, which the hair follicle actually produces itself, we had discovered that, then they can stimulate the development of mature mast cells from resident local progenitor mast cells. And cannabinoids, endocannabinoids as well as exocannabinoids, so THC, do the opposite. And they do this, both CRH and the cannabinoids, by impacting on the release of a critical mast cell differentiation factor called SCF, stem cell factor. The follicle makes it. And with this, it regulates. Do I want more mast cells? Do I want less mast cells? How do I want them to behave? So the follicle seems to be in control of what these mast cells do. And it bakes itself new mast cells, if you want. So if we then see that something is going on here in androgenetic alopecia with mast cells, their number increases, they become more active, we cannot ignore this, no? But it's being ignored. And I strongly suggest to put serious effort into dissecting this rigorously, because that's one cell population we know how to manipulate clinically already.
Right.
Yeah.
And so just to follow on to that, Ralf, is it, in your opinion, is it more about number of mast cells or mast cell behavior as its impact on hair loss?
Excellent question, Edward. I think it's both. So you can have too much of a good thing, right? But what shocked me most is when we looked at alopecia areata, so I'm switching back now to an inflammatory condition, that I was not really expecting. I always thought, I was in my residency training in dermatology, all I thought about if it came to mast cells was they are inflammatory cells, right? They degranulate and you get inflammation, boom. You get these hives, you get hay fever, you get asthma. But no, apparently, under physiological conditions, they are not pro-inflammatory. They're immune inhibitory cells. They probably support the immune privilege of the hair follicle from the outside, from the connective tissue sheath. And then something goes wrong, and they switch into a completely different phenotype. So their function changes, and they become aggressively pro-inflammatory. So the answer to your question is, we need to figure out, is there in androgenetic alopecia such a switch from a benign anti-inflammatory phenotype into a pro-inflammatory one? Nobody has done that. But the very fact that Kligman already found them to degranulate more, he used very primitive techniques, so now we have much better techniques looking at this, already makes you suspect that they do change their function. And then on top of that, if their number increases, for example, because in androgenetic alopecia the follicle sends the wrong signals to make more mast cells around it from progenitor cells, then it's good for it. then we would have an additional indication that something is going badly wrong here.
Got you. Max, do you have a question real quick or no?
No.
Okay. Well, why don't we? I think that was a nice.
Max is bored by mast cells already.
No. I was more thinking if we could study it, what would it be? But I'm thinking maybe we should keep that offline for now, or put.
No, it's very simple. I think what we really should do. We need to look back at this old Kligman study and see. So if we would do the study again, what else would we want to study? Which additional markers? How carefully would we want to look at what really is happening there? And so I think he made step 1. Now we have to make step 2, 3, 4, and 5 in defining exactly what is happening. So we have to basically scout the war theater, if you want, right? Then we have the opportunity to take scalp skin, healthy scalp skin, androgenetic alopecia scalp skin, and manipulate mast cells in it, and as selectively as possible, and then see how does the follicle respond. Does it matter? We bring them to degranulation, we make more mast cells, we make less. In the skin itself, in organ culture, the absence of bone marrow and blood, and then we see how does the hair follicle respond. And the most important thing we initially want to clarify is whether what Andrew asked about is actually happening, whether they change their function in androgenetic alopecia, and how do they change their function. And then we need to look back into our drawer of available drugs that target mast cells and see which of those could be used, and which of those could even be topically used.
Right. Okay, cool. So yeah, I mean, sorry, Max, do you have another one?
No, I think it's definitely something people are very interested in. So yeah, well, yeah. I wonder.
I give you one final thing on this to further stimulate excitement in finally dissecting the role of mast cells in androgenetic alopecia rigorously. There are patients I remember from my hair clinics with androgenetic alopecia, and that actually also goes for female pattern hair loss, who seemed to sense when they were getting the next flare of their hair loss. So androgenetic alopecia often goes in waves. It's not in most patients not very linear. They have episodes when they lose more hair and episodes when they lose less hair. And some of my patients said, well, I know when an episode is coming when my skin starts itching. And that immediately makes you think, of course, does the mast cell have something to do with that, right? So if you have androgenetic alopecia and an itchy scalp and you're still not interested in mast cells, you cannot be helped.
Good to know. Good to know. Back, stop touching your scalp, man.
I'm doing this already right now.
Yeah, right.
I have to scratch.
Gotta enjoy it while it's there.
Okay, well, Ralf, that was awesome. How about we riff a bit on? I think the whole discussion of epigenetic versus genetic role in hair loss, particularly because I know epigenetics in longevity have become really popularized by researchers like David Sinclair and things like that. I think there's so much overlap in overall longevity and longevity of the hair follicle. And I think just in our conversation earlier, as so many things in hair loss, I was kind of just fascinated by what an unexplored area of the field it is. Everybody likes to send in genes to the community, and the discussions of epigenetics are much more rare, and for good reason, because as you said, the research doesn't exist really as much as we would like. So yeah, if we could go there.
Yeah, so Andrew, this is an incredibly important discussion we're having now. I think one of the best things in these discussions we have here is that we can also define wide open frontiers where we must invest more research. And so the dermatology in general has not invested a lot of energy, clinical dermatology, into understanding the epigenetics of skin diseases. So this is a general trend. We are basically behind where the field should be, in general, in human skin research. However, our friends in mouse biology are way ahead of us there, and they particularly work out of the lab of Elaine Fuchs, have very clearly shown how critically important epigenetic mechanisms are in the biology of the mouse hair follicle. Now, what we don't really know is how much of this is really transferable to the human hair follicle. And in human hair research, there is a terrible scarcity of information on what are the key epigenetic mechanisms that we already know to be important in mice. Do they operate in the human follicle? It's almost 100% certain that they do, but we just lack basic information here, and it's critically important. If it is true that 70 to maybe 90%, depending on context and tissue, of the behavior of cells within a tissue is actually epigenetically regulated rather than genetically coded, then this is almost criminal that we have not put more effort into this. But we need to do this very systematically, and we also need to do this with the expectation in mind that it will take quite a while until we then develop drugs that can impact on this. For example, in epigenetics, drugs that have been already looked at more carefully, so-called HDAC inhibitors, have caused a lot of excitement in cancer research, but they are also loaded with side effects that you don't want, particularly not for a hair growth disorder. So finding drugs that interfere with epigenetic mechanisms that have a perfect safety, efficacy ratio will not be easy at all, and that then selective enough impact on those epigenetic mechanisms you really want to target and that really matter for androgenetic alopecia, all that will not at all be trivial. And we're talking about years of work here, but somebody got to start that. And this systematically and ideally in combination with human hair, between human hair biologists, clinicians, and mouse biologists with lots of expertise in epigenetics already. So that would be the ideal consortium. This should be a consortial effort where an interdisciplinary team attacks this always with one clear goal in mind. What does it mean for androgenetic alopecia, right? So that your community remains interested and invested. But I think it's absolutely necessary. And maybe one final comment on this. So the hormones that we know play a critical role in androgenetic alopecia, both male pattern and also female pattern hair loss, androgens, estrogens, prolactin, and several stress hormones, all are known to have dramatic epigenetic effects, but we have not put this together in an androgenetic alopecia context yet, and it's about time to do so. And to come back to what Andrew said in the beginning, we know that androgenetic alopecia and aging share some features, particularly in the dermopapillar compartment, but probably also elsewhere. And knowing how regulated aging is by epigenetic phenomena, it would be stupid not to look at this in androgenetic alopecia as well. So I think you're right on target, but you need to brace yourself for a huge effort if you want to do this seriously.
Mm-hmm.
Yeah. I think a big overlap with epigenetics is, excuse me, cellular senescence. And
yes, exactly. That's it. That's it.
How do you think about that, Ralph, between how many cells are dying in the dermapapilla or mesenchyme compartment and how many of them are senescing or becoming senescent?
Well, in the dermapilla, cells are not really dying. So, first of all, the senescence data we have on dermapilla fibroblasts from androgenetic alopecia hair follicles, usually, from what I remember, are senescence data in cell culture. We still need to find what happens really in the follicle, in the living scalp, while it is being miniaturized. What senescence phenomena really happen inside the dermapapilla itself? Because once you rip these dermapilla cells out of their tissue context and you culture them, all sorts of artifacts move in, which cloud the picture. So I would not rely so much on that. Hardcore markers for senescence being upregulated in the dermapilla during androgenetic alopecia hair follicle miniaturization still need to be published. There are first indications which suggest that yes, this does happen, but this has to be studied much more rigorously. We just had a little club of hair aficionados talking about this in Spain, Los Pilíferos, and we all agreed that's exactly what we need to do, and we discussed for a long time what are the markers we should be looking at. So we are ready to do this work. Death doesn't seem to happen much in the dermapapilla fibroblast compartment, but they move out. They emigrate from the dermapapilla excessively and don't move enough back in during the next anagen phase. So it's more a migratory problem, that the dermapilla compartment shrinks and shrinks and shrinks in size and in morphogenic potency by the number of cells getting less, but not by death, but by emigration, by excessive emigration and insufficient re-immigration during the next anagen phase. And to which extent then senescence of individual dermapilla cells or cells in the proximal connective tissue sheath comes on top of that remains to be seen.
Hey,
So-
go for it.
I was just going to ask, so because when those dermapapilla cells emigrate out of the compartment, they're going to the dermal sheath, right?
Yes.
So
Then
is
they stay there, and then they come back during early anagen.
right? So would we need to look at whether essentially what's happening to them after? We need to track them as they go into the dermal sheath, and then look what happens while they're in the sheath during catagen or telogen. Are they dying? Are they going out even
Yes.
somewhere else? Yeah.
Yes, exactly. That's what we should be doing. They're probably not dying, but so one of the worrying things about senescent cells is that they begin to develop this senescence-associated secretory phenotype. It takes a while to come up with it. Senescence-associated secretory. Anyway, so. They start secreting, for example, pro-inflammatory cytokines and chemokines, which then begin to impact on the environment around them, like little time bombs. They're sitting there and impacting negatively on their environment. Similar argument has been made for melanocytes in the epidermis, that senescent melanocytes big time contribute to aging of the epidermis. So if that were true also for senescent individuals, you can just have very few of these senescent cells. They can cause a lot of trouble by changing the signaling milieu around them that makes everyone else around them age faster too. So that's what we would be looking for. Are there indications that there are senescent cells that first of all express the right senescence markers?
Mm-hmm.
Then we look, what happens around them? Are they impacting negatively on their environment so that the other dermapilla cells, which were doing pretty well until then, are no longer secreting the morphogens that the hair follicle requires to stay large, big, happy, and in energy?
Right. Okay. And then Ralph, you had mentioned something earlier, and I had to touch on it because shout out Herbert. This is a direct community question. that we've gotten. But so you mentioned, you extract human scalp skin and you're testing something in vitro, and it's not the same as in vivo. What would you sort of characterize as like the biggest differences, or what might make scalp skin in vitro hard to translate into exactly what would happen in vivo? Like what are the biggest difference that sort of like cloud those results, if you will?
Okay, but let's. This is a very important question. So let's not confuse a few things. So my comment referred to cells that you extract out of the hair follicle and then culture in isolation.
Gotcha.
So cell culture of hair follicle, of individual hair follicle cells. Let me make a very nasty statement on this that may get me into trouble with several people. All research results generated with isolated hair follicle cells in culture are meaningless until otherwise proven. And that's a nasty statement. And the reason simply is that if you take dermopillar fibroblasts, you rip them out of their tissue context, they become like normal dermal fibroblasts very quickly. They change their properties. And the same you can say for melanocytes, the same you can say for keratinocytes. Hair follicle cells remain hair follicle cells as long as they are in a carefully orchestrated 3D context. You rip that apart, you are in deep shit trouble. That also refers to some extent to single cell RNA sequencing result, where you have to isolate these cells, you disperse them from each other with enzymes, which then immediately changes their gene expression pattern. So whether these cells really then do in vivo the same as they do in this cell suspension, gene expression assays, is anyone's guess. And then there is these 3D constructs where you try to partially, you first rip these cells apart from each other out of the hair follicle, and then you partially put them back together, hair folliculoids, or whatever you want to call them. They are a little bit closer to reality than isolated cells. So isolated cells are the worst, can't get worse. 3D is the second worst, because there are always some cell populations missing matrix components, are missing the exact architecture, in which the cells normally interact is very difficult to reproduce in these 3D systems. And of course, why would we even use them if we can use the hair follicle itself? So my argument is, if you want to make good preclinical research, use the ex vivo, not the in vitro system, which means you take the organ, you microdissect it, or you take the entire scalp skin with everything around it, and interrogate that experimentally. But even that, of course, is missing something. What is it missing? Well, it's missing normal perfusion. It still has some blood vessels, and they survive for a while, and even angiogenesis can occur. We've shown that for a number of days, but it's not perfused. These blood vessels are no longer functional, and of course, the hair follicles and the scalp skin that you organ culture has been severed from their sensory nerves. That's another limitation. But we have developed an assay where you can re-innovate scalp skin in the laboratory with the human iPS cell derived neuronal progenitors, and then you can get pretty normal at least, re-innovation phenomena. So even that can be covered, all under ex vivo conditions, preclinically in the laboratory. And if you're still not happy with this, then you can take. You say, hey, it's not good enough. Then you can take a piece of human scalp skin and you can transplant it on a mouse that cannot reject it. An immunocompromised mouse such as skid beige mice. And for that we have developed a beautiful androgenetic alopecia model together with Amos Gilha in Haifa, the Technion Israel Institute, where you then can, over many months, on a live animal that innervates and reperfuses this human skin xenotransplant beautifully within weeks, then you can study directly in the human target organ on a living animal what your test agent does. And we have already shown that androgenetic alopecia scalp in these mice, that's a real in vivo assay. What they do and how freresol minoxidil, PRP, finasteride, so the standard therapeutics, are impacting on both prolonging anagen and reversing in part the miniaturization of hair follicles from vellus to tuberous. So all that can be studied. And only then you go to the patient, which of course always is the ultimate test. And often enough, what you have shown before cannot be entirely translated to the human situation. But if you have run through these steps, you begin with microdissected hair follicles and or right away scalp skin organ culture. And our experience is if you see phenomena in these target organ cells that have been manipulated as little as possible and are as close to the real thing as you possibly can get in a laboratory setting, then the chances that what you will find there, the responses you will find there, really reflect what you will get on a living human being are as high as humanly possible.
Gotcha.
Beautiful. Yeah.
Yeah. I had a cool another topic, but sorry, Bax.
I was just going to make a bad joke. Just there's a running joke right now, Ralf, that you can sell anything to patients to test in vivo as long as you put for research use only on the website. So if
yeah, that's a bad joke indeed, because it replaces animal testing, and it makes basically it creates the illusion that the kind of work that people like me are doing is superfluous. Now, why even doing all this research? Just slap that shit on the human scalp, no? But if you lose a few hair follicles, or you look funny after you do that, or you feel kind of funny, well, that happens. Yeah, if that's your philosophy, I'm not your man.
well, you'll be surprised. The people who do this, they love you very much. I think. People are just urgent. Yeah.
I understand, and it's perfectly human to want results fast and to avoid all these levels of potentially artifactual results, right? So if we avoid cell culture, we're already doing a good job. We avoid generating data that may be completely meaningless or even misleading. And I would also avoid 3D models whenever possible. There are some very specific reasons when 3D models are useful. If you want to, for example, genetically manipulate a cell population so that it responds in a highly defined manner to experimental stimulation, and then put it into a 3D context. So that's a good reason for using 3D, but that's, I think, the only really convincing one. Otherwise, go straight for the organ. And if you do that, I think it's always well advised to have a bit of patience and see, is that stuff I want to put on my scalp really doing at least a bit of what I'm hoping it to do in these organ culture systems? If it does, then I think you are much better advised to put it on your scalp. And that's another reason why you should be careful with this. Hair follicles and scalp skin are a beautiful toxicology early warning system. The follicle is so delicate in its growth and is so demanding and so responsive to all sorts of things. It will also reveal potential toxicity, side effects of what could happen if you put this on your own skin. And the same goes for scalp skin. So these organ culture systems are very sensitive systems. Not only do they tell you, do I get positive or negative hair growth effects, but also they anticipate potential side effects, and to not use this screening step before you dump this stuff on yourself is dumb.
Agreed.
Great.
So Ralph, I have a kind of a I have a cool topic that we'll work in real quick, and bear in mind. So this is related to CosmeRNA, but we're not going to go too deeply into CosmeRNA because again, I think it warrants more deeper look. But I should give you some context. So we had a tweet where effectively calling CosmeRNA a scam. And the scam, the scam language is probably was a little bit too extreme, and people, it was a little bit spicy. People got a little bit upset with us because scam is probably not the right word. But to give you a little bit of context, right? So CosmeRNA uses siRNA to effectively suppress the androgen receptors downstream of DHT. It's now being sold on Amazon as a cosmeceutical, which I know you were pretty shocked, right? That an siRNA. Yeah. So that was kind of interesting. But I guess where sort of like the scam language came from was we felt like they were perhaps manipulating or presenting their data in interesting ways to maybe mislead the hair loss community, that this could be a more effective treatment than not. People were quite excited because its side effect profile was really good. So they at least no observed side effects yet. And then I think what they showed, they tested a low dosage of like 0.5 milligrams per milliliter and 5 milligrams per milliliter. They tested topically. And they tested for the 5 milligrams dosage. They were testing once a week application. They're selling it as once every 2 weeks. So right there, you're probably going to have less efficacy. And I think the other thing they did to sort of manipulate their data, not manipulate the data, but in comparing it to finasteride, they said, okay, so their solution of once every week 5 milligrams resulted in a hair count growth of like 7 hairs per square centimeter, something like that. And then they used the finasteride comparison of 9.3 hairs per square centimeter, which is one of the lower ranges of like finasteride regrowth. So they kind of like understated finasteride's effect, and then also presented their results if you're applying, their solution like twice as much as they're prescribing it on the open markets. So we don't need to go too much into
So did they even use the same methodology to count the hairs? A classical Lega artist performed photo trichogram and vertex global photography. Did they do that?
We have to confirm, but I think so.
That's always critical. So if you compare two data sets, if they have not been generated with the exact same methodology, how to photograph, and you remember the phototrichogram requires that you first shave an area, then you see which hair shafts are growing, then you rephotograph, and then you do this. And the global vertex photography requires that a panel of independent blinded observers gives you an impression of is there an impact on hair growth improvement or not. So if they have not done the exact same methodology, one cannot compare those data.
Right. And so I guess where I wanted to go with this is, everybody in our community is reading a ton of studies. And I know we've talked about it a bit offline, like just how the various manipulation tactics that people will use when presenting their data. Like this sort of taking advantage of like seasonal hair regrowth and things like that.
Yes.
To CosmeRNA's credit, the placebo group actually had a slight decrease in target area hair count. But maybe if we could just riff on that a little bit, what people should be on the lookout for.
Well, just to add on real quick, sorry, we'll read studies where there's some new treatment called hair stimulating complex, right? That came out in 2011. No one's ever heard of it. Nothing ever became of it. But it generated, 11 hairs per square centimeter growth in their, double-blind placebo. So which is obviously better than CosmeRNA's, and even on the low end of finasteride. Obviously, this treatment hasn't shown any efficacy in the market. So yeah, and even with melatonin, like other treatments too, you'll see this frequently where there are good lab results, but then people will try it and effectively get nothing.
Yeah, it all depends on what you measure and how you measure, and how long you measure. So the problem with clinical hair trials is that cosmetic industry is always in a hurry, right? So they rarely run a trial longer than 3 months. The minimal duration of a good hair trial of any kind that tries to be hair stimulatory, it's different for hair growth inhibitory compounds, should be 6 months. And if any trial that's already shorter than that is questionable per se. I've been involved also in trials where we did it shorter for budgetary reasons, but only, and this is one has to give credit to the when finasteride was first investigated carefully. They basically set the gold standard for how to do it right. The gold standard clearly now is you do it 6 months. You do phototrichogram, not just a trichogram, a classical phototrichogram, which requires that your patient comes in first, being shaved, photograph taken, comes back in 2-3 days later, photographed again, then comes back, let's say after 3 months and after 6 months. And all this is very objectively counted then. Ideally by using machine learning techniques, right? There are now very objective ways of doing this. Previously it was counted by hand. Now, if you have the right computer program, you can have much more accurate assessments. That also then allows you to assess have there been any changes in hair shaft diameter, if the program is good enough. Then, however, phototrichogram does not really give you. It gives you a great idea. Has the percentage of anagen to telogen versus catagen follicles, so the last two you can't distinguish, but with this technique, changed. And that gives you a hair cycle response, right? If you're lucky, you can also get an indication, was the hair shaft diameter improved a little bit? And if you're extremely lucky, and that's where most systems, Canfield, Tricoscan or so often fail, has the percentage of vellus follicles changed? And that's for that actually, if you want to get 100% certain results, you really need a biopsy, because then you need to by morphological criteria in a skin section, see has there been a vellus to terminal reconversion, which in fact has been disclaimed to even happen by one group of researchers because they relied on phototrichogram results. We just showed in this xenotransplanted human skin on mice, you can study over a long time, that it does happen if you use histology. So the use of histology for some claims that are very important, vellus to terminal reconversion, is a critical parameter that you want to be sure whether your test compound is doing it. For that you need histology, and that's almost never done, right? Mm-hmm. And then finally you need this global vertex photography, because you look at this, I'm losing my hair here, right? There you need a standardized photography that checks overall what has been the cosmetic effect, and then you show these photos blinded to double-blinded actually to observers who say, well, improved, not improved, and give it a number. Then you have a trial, and if you want to be super perfect, after 6 months you switch the placebo group to verum and the verum to placebo. And if then the numbers changed as expected, then you know you're sitting on something that actually does something reproducible. And the final question always is, what is the minimal number of people that should be studied in both the control and the test group? And that's often very difficult to say because it depends on the expected difference you will see between test and control groups. But if you already knew the difference, biostatisticians always ask you when to calculate the number of minimal patients, the minimal number of patients you should enroll. So what is the difference you expect? Yeah, well, if I knew it, why would I run the tests? So you have to make gross estimates, and then it often turns out that the difference was smaller than you expected, and you had underpowered your study. So yes, you see a trend, but it's not really significant. So in a nutshell, the more people you can enroll, the better.
Gotcha. And then last question, just before we go next topic, was your surprise with the siRNA?
Sorry, let me just clarify this. So your initial question was, how do we know which data to trust?
Yeah.
Right? Initially, that's what you asked. And that's my answer. If they have not done a 6-month study, they haven't done a classical photodiagram, there's no global vertex photography, and no biopsy, be skeptical.
Yeah, CosmeRNA doesn't have the biopsy, and in their treatment group there were 22 patients, so definitely
No N, but could suffice if the difference was massive. And how long did they treat?
It was 6 months, I believe.
24 weeks.
Yeah.
Yeah, that's pretty good. That's pretty good.
Yeah.
Okay,
Yeah.
sorry, I was interrupting you.
No. So I was just, before we moved on to the next one, was your surprise with the siRNA being sold as a cosmeceutical? Like, are you expecting certain side effects, or is it just the fact that we just don't really know enough about what happened?
I've not heard of any siRNA being approved as a cosmetic agent, because with siRNA you of course substantially impact into the translation of proteins, in this case a receptor protein, right, from the transcript, and you also impact on transcription itself. That's genetic manipulation, and if we allow genetic manipulations to go through as cosmetics, I mean, where's the limit there, right? You got to draw the line somewhere. So I don't know how they got away with this, if they really did get away with this, and this was all legal.
Well, it was approved by the EU and UK, but our theory, so they never took biopsies, so we don't even know if the drug gets to the base of the hair follicle, right, when they apply it. So I mean, there are a lot of reasons to be skeptical, but yeah, we can move on, I think.
It was South Korea, right, that rejected it?
Yeah.
Let me say also something else. So nature put androgen receptors there for a reason, right? And we have androgen receptors in our skin for good reasons, and they are part of our skin physiology. We just don't understand enough what are various compartments, various cell populations in our skin really doing with all these receptors. Androgen receptors, they all express androgen receptors at different levels, but they all do. There's hardly any cell that does not express androgen receptors. So if we start blocking them indiscriminately in all cells, right? Okay, one could say those cells that express most of them, let's say dermal pillar fibroblasts, will be most sensitive. Yes, but what about if your hair follicle epithelial stem cells also need a little bit of stimulation via androgen receptors? And I block this for years to come. Is that going to be a good thing? How does that change the metabolism of the skin? What side effects will I get in the long run if I indiscriminatively block androgen receptors or reduce their signaling functions? Is that really a good idea? I'm nervous about this, at least, and I would want to see a lot more toxicology studies, a lot more safety work done before this is administered to human skin.
Yep, makes sense. Makes sense. And Bax, you had a topic you wanted to wrap it up with?
Oh, well, I was just thinking, Ralph, to wrap up, maybe because I actually read your Prolactin 2006 paper. I think you were the first one to discover that it's made inside the follicle.
Well, Well, I think there was also an Australian group that had shown this in the mouse, and we had, but in the human follicle, yes.
Yeah, you were the first one.
And actually, now that you remind me, at that time, my close friend and collaborator, Andres Slominski, had also looked at it, and he said, no, it's not there. But he had looked at the wrong part of the skin, because it's very prominently there in scalp hair follicles when they grow.
Yeah.
Sorry for interrupting.
No, not interrupting at all. So I was just curious, because obviously 2006 was a little time ago, just like, why you either didn't pursue it further, what red flags you saw, what you liked about it, just your overall.
Funding. It's always the same story. So I think that prolactin story is super interesting, but we got confused in the beginning. If I remember correctly, and I hope I'm not mixing this up here in public now. Initially, we looked at male hair follicles and male scalp hair follicles, and I think they were from frontotemporal skin. No. They were occipital male hair follicles, and there we found hair growth inhibition by prolactin. We found that you not only did they make prolactin and they expressed the receptor for it, but if you stimulated anagen follicles with prolactin, they were kicked into keratin. Then we repeated the same thing with frontotemporal hair follicles from females after facelift surgery, and we couldn't get this work repeated. On the contrary, we found the opposite. Their prolactin stimulated hair follicles. They stayed longer in anagen. So we published that and thought, well, there's this big gender gap and we've got to be very careful. But we never really figured out, and that's something I need to do. So was it a location difference or a gender difference that caused this completely opposite, diametrically opposed response to the same hormone? I think it's primarily a gender dependent difference. But that tells us that some of these hormones, how the follicle responds to it is entirely gender dependent. And I expect that something similar also happens for other hormones. We have very first pilot data. We just found that growth hormone is a hair growth inhibitor, despite its name. And it's also made in the human follicle. But again, we studied, in this case, at first in females, where it was hair growth inhibitory. We have one sample of male hair follicles where we think it's inhibitory. So we have confirmed all of that, but it makes me acutely aware of the necessity, whenever you investigate any hormone whatsoever, that you very carefully not only standardize from where you take the hair follicles, let's say frontotemporal, vertex, occipital, but also standardize for gender. And we may even have to standardize for ethnicity. How did we get into this?
Prolactin.
Prolactin. Yes, and then, and the final thing, that's the last paper I think we seriously did on this. We then found that prolactin actually in the hair follicle stem cells, so in the bulbs, the epithelial stem cells that help regenerate the follicle during each cycle, they depend on prolactin. So they really need it. They need to be stimulated. If you stimulate the prolactin receptor there, you do not want to inhibit it. You want to stimulate it because these stem cells are happy. So if you used an antagonist against the stem cell, the prolactin receptor, it might be good for hair growth of male scalp hair follicles, for their cycling, might keep them longer in anagen, but it might be bad for their stem cells. And in females, it should be bad all the time. If our work, ex vivo work, translates into the clinical situation.
So this is something we might even have to look at, like the female-male difference with the thyroid hormone study, for example.
Yeah, that's possible. Absolutely. I'm not so much worried about thyroid hormones because the gender dependent differences in thyroid hormone responses are not so dramatic, at least according to the literature. For prolactin and for androgens in any case, there have already been lots of other studies in animals, in cell culture, that suggest gender-dependent differences are important. Not so much for thyroid hormone. So while in principle you are right, I wouldn't so much worry about that there.
Good for us, I guess. Okay, well, Ralf, I mean, this has been incredible. If I don't know if any you guys have anything else you want to touch on, I think this was a pretty great impromptu episode, and we'll dig in a little bit deeper next time to the reviewed by Ralf topics.
I think we almost even hit all of them, to be honest, besides PI1 inhibition.
Interesting, Ralf. I'll give Jumpman some flowers here. One of our sort of top community researchers actually found that PI1 inhibition for a bunch of reasons could be good for hair loss, but actually what he found is that T3, T4 thyroid hormones might actually inhibit PAI1. So there's cool overlap with what we're doing right now.
That is very nice. Well, so I'm gonna write up a little bit, a few comments on these 3 projects you had sent me, so give me at least a week, and then we can continue this discussion if you still are interested.
Amazing.
We'll be interested. There are a lot of people who will be interested.
We will be interested.
But hey, guys, you came up with this. Mast cells need to be investigated very carefully, right? In androgenetic alopecia, I fully agree. And let's also put macrophages in there.
Yeah.
They are also underestimated in their hair growth regulatory importance. So we've done a lot of work on this ourselves, both in mice and in human scalp. I applaud you for pushing the epigenetics angle in androgenetic alopecia research. Let's follow up on this and get serious about it. Let's put together a consortium of just the right mix of interdisciplinary researchers to finally address this, because I think it could be giving us completely novel targets that for therapeutic intervention that we have not even carefully considered, but that are totally important, and that some of the hormones we just talked about are already using.
Mm-hmm. Oh yeah.
Yeah, we can reach out to the VitaDAO community. I know you've done some work with rapamycin, and but they're obviously looking a lot at epigenetics. So hopefully, maybe we get some economies of scale. Use the human hair follicles as our model. That'd be pretty exciting.
The first thing you need is funding. The second thing is, once you know how much funding is there available, then one thinks what would be a good team to work on this together. Mm-hmm. Worry about funding first.
Yep.
Gotcha.
We are. Yeah, I guess that's it. With that, this has been the 3rd episode of the Hairy Matters podcast. Hope you guys enjoyed it, and we'll be back soon next time.